Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning sessions. Today in session 4 we learn about Fischer projection and Haworth structure of few monosaccharides. In your syllabus, Fischer and Haworth structure of few monosaccharides has been included. First we'll take up Fischer projection. This was devised by Emil Fischer in 1891. It's a two-dimensional representation of an organic molecule. It's a two-dimensional representation of the organic molecule. And here Emil Fischer represented the two-dimensional structure in the form of projections on the carbon skeleton. On the linear carbon skeleton, he represented the functional groups in the form of the projections. And this in honor of Fischer, Emil Fischer, it was called as the Fischer projection. First we'll take up the Fischer projection of an aldose pentose, that is the ribose. This ribose is having the formula C5H10 and O5. It is having C5H10O5. This ribose is very important in the structure of DNA and RNA. DNA means deoxyribonucleic acid and the RNA is the ribonucleic acid. This ribose sugar play an important role in differentiating nucleic acid either as DNA or as the RNA. The ribose sugar is present only in the RNA, but in case of the DNA, this ribose sugar will not have the oxygen atom at the C2 position. Oxygen atom at the C2 position. That ribose molecule without the oxygen is called as deoxyribose sugar. This deoxyribose sugar will be present in the DNA. DNA will be having the deoxyribose sugar, whereas RNA will be having the ribose sugar. This ribose having C5H10O5 formula, and here it has this structural formula like here in a Fischer projection, the carbonyl groups like CHO and CO will be placed either at C1 position or at C2 position. The ribose is having the structural formula CH2OH, CHOH, CHOH, CHOH and then CHO. And in Fischer projection, CHO it will be placed at the first carbon atom or the second carbon atom in case of ketosis. Since it is a ribose or since it is a aldose pentose, here the carbonyl group, aldehyde group is placed at the first position on the top of the projection structure. Then you have CHOH, that is the first carbon atom will be having the carbonyl group, second carbon atom is having OH group or the hydroxyl group towards the right side and H towards the left side, and at C3 position it has OH towards the right side and H towards the left side, just like our C2 carbon atom. And at C4 carbon atom we have again OH at right side and H at left side. At C5 carbon atom you have the 2 hydrogen atoms and 1 hydroxyl group, that is CH2OH. So here you can count it has 1 to 5 carbon atom with carbonyl group on the top of the structure, and it's an open structure where you have the projections of the functional groups on the carbon skeleton. So this is the structure of the D ribose. Haworth projection. Now we'll move on to Haworth projection. Haworth projection is a common way of writing structural formula to represent the cyclic structure, of monosaccharide with a simple three-dimensional perspective. Our Fischer projection is two-dimensional, but Haworth projection is three-dimensional, but it has the cyclic structure. It was named after the English chemist Sir Norman Haworth. Fischer projection was devised by Emil Fischer, and this Haworth projection was shown by Norman Haworth. And in honor of the Norman Haworth, the scientist, this projection is called Haworth projection. Here, carbon 1 is known as anomeric carbon atom. The first carbon is known as anomeric carbon, and the groups below the plane of the ring, the cyclic structure will be having the series of carbon atom, so it will be like this. Haworth structure will be like this, and it's a cyclic structure, and there will be cyclization, or the ring formation will be taking place between the first carbon atom or the fifth carbon atom, or this structure will be showing the three-dimensional structure, and on this we are writing the functional groups, and whatever the functional groups that are there below the Haworth structure, they represent the functional groups which are there on the right side of the Fischer projection. Whatever the Fischer projection will be like this. So on the right side, whatever the functional groups are there towards the right side, so they will be there on the downward side of the Haworth projection, and the left side functional groups will be there towards the inner structure of the Haworth projection. The cyclic forms of D-glucose are six-membered hemiacetals formed by an intramolecular reaction of the OH group at the C5 position with the aldehyde group. For example, in case of glucose, six-membered hemiacetals are formed by an intramolecular, that means the C1 of the glucose and the C5 OH and H will interact, and they result in the six-membered hemiacetal ring. So these cyclic structures of the monosaccharides are named according to their 5 or 6-membered rings. For example, in case of the fructose, this cyclization results in the formation of the 5-membered ring. So the 6-membered cyclic hemiacetal is called pyranose. 6-membered cyclic hemiacetal is called pyranose, derived from the name of the 6-membered cyclic ether, that is pyran. In case of the glucose, the cyclic structure will be formed between C1 and C5. At C1 we have the carbonyl group, and the C5 will be having the OH group. So this intramolecular interaction will result in 6-membered hemiacetal. But in case of the fructose, since we have the carbonyl ketone group at the C2 position, we will find the formation of the 5-membered cyclic hemiacetal, and this 5-membered cyclic hemiacetal is called furanose. So it derived its name from the name of the 5-membered cyclic ether, that is furan. 6-membered structure, Haworth cyclic structure is called pyranose, and 5-membered Haworth structure is called furanose. This is the Haworth structure or the Haworth projections of ribose. So earlier you saw how we write the ribose structure on the Fischer projection. This is the Haworth projection of the ribose. Here at C1 position you have, since there is the cyclic ring formation, you have the C1 atom will be having OH on the top and H on the below the structure of the Haworth structure. At the C2 position you have the H and OH. OH will be towards the downward side and H will be upside, whereas at C3 you have OH downwards and H upwards. And at the C4 position there is, you have H atom, and then 5th carbon atom, that is CH2OH, is placed. So this is the Haworth projection structure of the ribose. So this ribose is an important component of RNA. It is an important component of RNA, that is ribonucleic acid. Whereas in case of the deoxyribose, the DNA, in case of DNA will be having the deoxyribose, where you will not find the oxygen at the C2 position. You will not find the oxygen at the C2 position. So it is named as 2-deoxyribose. It is named as 2-deoxyribose, and it is having the C1 atom OH upwards and H downwards, and since at C2 oxygen is not there, you have the H upward as well as downward. But in case of C3 you have H upwards and OH downwards, and at C4 you have H downward and CH2OH, that is the 5th carbon atom, is placed. So here there is formation of the cyclic structure between the C1 atom and the C4 atom. C1 atom because it has the aldehyde group, and C4 will be sharing, intramolecular interaction takes place and results in the formation of the cyclic structure. This is regarding the Haworth and Fischer structure of the ribose. So this is very important as far as your examination is concerned. You should learn Fischer projection of each and every monosaccharide along with their Haworth structure. So in the next sessions I will be taking the Fischer and Haworth structure of remaining monosaccharide. I hope today you understood the Fischer and Haworth projections, how to write using the Fischer formula and the Haworth 3D structure. Okay, thank you.